Our newest paper, is out. Um,
Do you have to boast some more?
I have to boast a little bit. I am very proud of this one. So this is in the, I will link it. PNAS,
It
the
is very
Proceedings of
cool.
the Natural Academy of Sciences. Astrocyte-derived neurons provide excitatory input to the adult striatal circuitry. And this has been a project that we've worked on for, well, about 5-6 years. For various reasons. So it started kind of with a study back in 2014 that we were not involved in the lab of Jonas Friesen at the Karolinska, who showed that if you have astrocytes and you disable notch signaling, they will start a neurogenic program. So they will start to produce more new neurons. And the underlying idea is that, hey, perhaps if we can generate new neurons on demand, we can use that to alleviate symptoms in diseases like Parkinson's or Alzheimer's disease. So this was a
It's a
very exciting
cure
finding.
for all diseases.
I mean, if you can produce new neurons and they do the right thing, then
Yeah, of course, yeah.
any neurodegenerative disease will be you will be able to target. So, but they found they could create new neurons, and one of the ways you show that it's a neuron is so they expressed NUN, which is the marker for mature neurons. Um, but what they didn't show yet was whether those new neurons actually do anything. And that's what we were setting out to do. So we looked, I say we, Christian's lab started this, and they started looking at, first of all, where these neurons are. They found mostly on the medial side of the striatum. So the striatum brain area, subcortical area, very
So do you,
important.
do you target the whole striatum or the whole brain or like the
So the
whole
whole brain,
mouse?
the whole mouse technically.
So it's like a knockout of this notch thing. What did you
So
say?
it's a conditional notch signaling knockout, RBJK knockout specifically, um, which we can induce with tamoxifen. So we wait for the animals to become adults, and then we knock it out.
Okay, otherwise you will mess with the development of everything,
Yeah,
the
and
whole
we want
brain.
to see,
Yeah,
if this can happen
in adults.
in adults. That was
Mm.
the whole point. We want to see adult neurogenesis. And this works. So the mouse model has been developed by that previous study in the Friesen lab.
Ah, yeah.
And then Christian started look, okay, so most of these neurons, the new neurons, we find them in the striatum, in the medial striatum, next to the lateral ventricle. And he tried, testing what kind of neuron these were. So the typical neurons in the striatum, about 95% of them, are so-called medium spiny neurons. These are GABAergic neurons. They express GABA. They project down to the globus pallidus and the midbrain. Express DARP32, a few other markers, D1, D2 receptors, all that. So they tested the newly generated neurons for those, if they express any of those markers, and they didn't. So then they thought, well, okay, we'll test them for the interneurons in the striatum. So we test them for parvalbumin and somatostatin, and they don't express those either. So they tested them for acetylcholine, nNOS, a whole bunch of markers, and basically these new neurons didn't really express any of them. Wait, why can I not?
Can you not show us all the markers?
No, it's weird. I gotta here. Oh, now it works. So here you can see, see it sort of. So they don't express DARP32, they don't express chat, parvalbumin, calretinin, or somatostatin. These are really the most common markers found in the striatum, which surprised them very much. So we have new neurons, but they don't look like any of the other neurons in the area where they are. So what are they?
But he did establish there were neurons because they have this neuron marker, right? The
Yes.
neuronal marker,
Yes.
yes.
So they should be neurons, but we've never even tested if they could fire action potentials. So that's where we came in, because we do electrophysiology, and Christian approached us and said, hey, can we patch these neurons and look what they look like electrophysiologically? So it is where you put a patch pipette in, like a tiny little glass pipette, and then you record the electrical signals of that cell. Which turned out to be a pain in the ass. Because these new neurons are super duper tiny. Like here, the bottom one, that's a normal medium spiny neuron. And then these are like about half the size. It's a real pain in the ass to patch them. And they're super small. And it's very easy to kill them when you don't approach them just right. They're very sensitive.
Did it help if you pulled like smaller pipettes? Or?
Didn't try.
Oh, you didn't try. This is what I was thinking. Like if you have smaller cells, it might help.
The trouble with that is, of course, the smaller your pipette tip, the higher the resistance. So the less accurately you can record, and the more easily it gets blocked. I don't think it would really help you with patching.
Yeah, but you have a range, so you could sit on.
Yeah, there is a range, and we were sitting on the high end of that range.
Yeah, okay.
But you make your pipette much bigger, much smaller than that, and it will very easily become clogged, and then you will not have a good recording. So you have to make a trade-off, because if you have your pipette too big, it won't get clogged, but it will kill your cell. If it's too small, it won't kill your cell, but it might get clogged. So you want to be somewhere in that optimal range, which is not, you don't have a lot of play in either direction. But we managed to get to record, and lo and behold, these are actually neurons. They fire action potentials. So that's cool. So we know, okay, they're neurons. Are they anything like the rest of the cells in the striatum? So I did a whole bunch of electrophysiology. Maybe I should get the... where's the figure? Yeah, I can show this. I think this is allowed. So you can see the IV curves for these neurons. Astrocyte-derived neuron here in red. An astrocyte, of course, doesn't fire any action potentials. And then we have the MSNs, FSIs, chins, and LTS neurons. And basically they're not like anything, any other striatal cell. So the next question that we asked is, okay, so they're completely novel. They don't express any of the common markers. They don't look physiologically like striatal cells. Do they actually integrate into the striatal circuit, right? Do they receive input? Do they contact other neurons? Otherwise, they're just, freely floating cells that don't do anything.
But you could excite them.
Yeah. They can fire action potentials, but I don't know if they release neurotransmitter.
No. But then I'm thinking like, I'm not sure if a neuron that does not make any connections, if it survives and fires action potentials.
It does not. It does. So, or typically it gets pruned. That's
right. That's what. Okay,
so we expected to find something, and so we zapped the slice. Basically, you put a big electrode next to your cells. You zap it with electricity, so all of the cells around you fire, and then we look for input, and we found input. So this confirms they do receive synaptic input, which you can block with GABAzine. As I said, most of the neurons in striatum are GABAergic, so they release GABA, and if we block GABA, we don't see input. So this all makes sense. They do receive input from nearby cells. The next we wanted to know, so if they receive input, do they also produce output? Do they connect to anything? So I tried patching a lot of pairs and triplets. You have one astrocyte-derived neuron and then maybe two other neurons nearby, which did not go well. Turns out, these neurons are really difficult to patch and they very easily die. And if you, want to stick your electrode in the cell, in the slice, and then another electrode nearby, the whole slice moves. And after a lot of sweat and tears and, a lot of swearing, I eventually got a handful of pairs and they were not connected. So...
Yeah, because if they're not highly interconnected, you don't have such a good chance of getting a connection in pair.
It's not a high chance to begin with. Yeah. So we took a different approach, and we asked Christian, and they were able to cross these mice with a channel rhodopsin reporter. So it's a light-gated opsin. You shine a light on these cells, and they will fire action potentials, which is way easier. And we did that. I can show the construct. So we did that. And then we recorded from the postsynaptic neurons
And
to
the
look if they
postsynaptic,
received anything.
the
And
postsynaptic neurons, they're just any neurons
any
in the
neurons, mostly
same
MSNs.
vicinity. Or...
Yep. And it turns out they do. It totally worked. We saw postsynaptic responses in MSNs when we shine light. And here's where it got really interesting. So we tried blocking those responses with GABA, because we thought, well, all the cells in the striatum are basically GABAergic, except for chins. And it didn't work. And then I blocked those. Then I tried to block them with MBQX, so which blocks, you know it.
AMPA? What?
Yes.
Are you asking me? Are you doing an exam for me?
So glutamate connections. And that worked. That totally worked. So it turns out these neurons use glutamate as their neurotransmitter. Like they're the only cell type in the striatum that uses glutamate. It still blows my mind.
So you didn't try AP5? What is it?
I tried DAP5, yeah.
AP5
yeah.
for NMDA.
Yeah. There's a small residual component that was blocked with AP5. It was I only had like a handful of cells, so I don't know if we ever got that statistically significant. I don't remember. But we did that, and yes, there was a small residual component that disappeared with AP5. So they have there's AMPA and MBA receptors. And this makes sense because MSNs receive very strong cortical input. So they receive a lot of glutamatergic input from the cortex. So it's normal that they would have glutamate receptors. Anyway, because now we knew that these neurons used glutamate, we could test for glutamate markers, which we did, and they totally do.
Okay.
So these new neurons express vGlut1, so the vesicular glutamate 1 transporter.
Wait, why didn't they test for that before?
Because it's in the striatum. There's not supposed to be any glutamatergic neurons in the striatum.
Okay.
We also tested for vGlut2. We didn't detect any of that, but they express vGlut1. So yeah, so this is a completely new population of neurons that are produced by astrocytes and use glutamate to innervate other neurons in the striatum, which I think is super cool. And it's now out. It took a long time to get this published, but we made it in the end. If anyone wants to know more about it, hit me up.